But yeah, so I joined WikiPathways in 2012, but the project has been around for longer. The project is a collaboration between our group and Chris Avelo in our group and Martina Koetman. They're the Maastricht part, central part of the architects, as they're formally called. And in the United States, this is the group of Alex Pico at the Gladstone Institutes associated with the University of California in San Francisco. And my background is chemometrics. I actually got my PhD in a group of Lutgarde Buidens in the Netherlands. But I also have a cheminformatics background, which puts me really on this edge of chemical information, structured databases, and how do we use this in extracting knowledge about this. And over time, metabolomics and lipidomics, or lipidomics actually came a bit later, but the area of metabolomics and omics data, in the Netherlands Metabolomics Center, for example, I slowly moved into towards more and more bioinformatics and data integration in that area, and that you will see reflected in this presentation. So basically, what my interest in is getting more information together about the biology that we're studying. And our experiments, they give a lot of information, but it's still a bit isolated. And we want to basically link this to information that we know from other sources. Just like this last question, for example, this protein is not found in this tissue. So how do we get all that information together? And that's basically the integrative systems biology that our bioinformatics, that our systems biology group focuses on. So from my side, there are two databases that have been connecting here. One is WikiPathways, the other is Wikidata. And Wikidata came in because we actually needed more metabolite and later also lipid identifiers, because we saw a number of databases mentioned: PubChem, HMDB, LIPID MAPS itself, of course, CABI, but all these databases, they have their own characteristics and therefore their own limitations. And we needed something more general, and we found, Denise and I found that in Wikidata. But then Wikidata actually is a project about 7 years old now. We'll see that it's getting really powerful. So this is where Wikidata becomes a central resource in actually connecting all those databases. And I hope my slides will give some idea about that. But we'll start with WikiPathways. In the middle of the session, we already actually saw one of the WikiPathways. I copy-pasted the link in the chat, one of the lipid pathways. But the most recent paper about WikiPathways is this one. And this last NAR article about WikiPathways focuses particularly on how we're connecting communities, and LIPID MAPS is one of these communities. But we have many more recent portals, recent communities being set up are, for example, about Daphnia magna as a new model system where there is not a lot of pathway knowledge available yet. We can homology model, if you like, pathways, like your people tend to do with CAC pathways as well. But if we really make them species specific, we really can get species specific information in there. This does extend to, and I don't have a slide for that, but that was also why I liked that last question, is that we had a project for making these pathways tissue specific, and that's a tissue analyzer, and I'll look up and link to that later on. That has not been published yet, so I can't point to a paper at this moment. But pretty much like the other presentations this afternoon, I mean today at least, we saw things like we have our experimental data, we try to make sense of that. And so this week you've seen a number of those approaches. Today we saw enrichment approaches, for example, for lipid classes. And the same kind of approach is what you do in WikiPathways as well. In this case, as common with a pathway analysis, you try to find which biological processes, which biological pathways are relevant to the experimental data that you have found. And now this can be a composition of membranes, as we saw earlier today, for example, or what we see here. This is metabolomics, not lipidomics so much, but the idea is the same: a pathway where we see something happening consistent with the data. Actually, sort of like what we saw in BioPen in the previous presentation, but here based on existing knowledge. As in, yeah, reactions with a lot of information to that, the full pathway, etc. So that is one thing. If we do not necessarily know the exact thing, so if you do sort of a correlation analysis, or you want to bunch together a whole set of pathways, that is another approach that our group has been taking. Then you go to Cytoscape and you start stopping doing the pathway analysis, but doing the network analysis. And that is an interesting thing, because then you can really start pulling in additional information, with site target linker, for example, and in this case a bit aimed at pharmacology, linking here pathway information to drugs and diseases. The drugs here, the compounds in pink, purplish, and diseases in blue. But similarly, we can add then information about transcription factors, methylation states, and sorts, and pull in all that kind of information to really get well, by combining more and more data, looking into the same problem, getting them better and better insight in all the biology that is happening there. Now, these pathways, they have to be drawn, they have to be created, they so I just asked, so for BioPen, for example, can we export those networks that we get there to have them as seed as a pathway in WikiPathways, for example? And so the tool developed in our group for drawing these pathways, as well as actually mapping experimental data on those pathways, resulting in the in plots like this, where we, well, in this case, a bit of metabolomics, experimental metabolomics data, fold changes, like in the other presentations, on top of these on these pathways. Same thing you can do. Sorry. Same thing you can do in Cytoscape. You can map this experimental data on top of it, but the drawing is done by Physio. This is our pathway editor, and this is some reference literature about that. The bottom right articles, actually, how you can automate this, how you can interactively work with these pathways as well, and we'll actually see that a lot has happened there since that publication, actually about 6 years ago. So this is what PubPhysio looks like, annotated with blue comments and red comments, indicating some of the information that we have in the pathways and WikiPathways. So the enzymatic transformations are in there, in drawn here, which enzyme is involved there. Each of the metabolites or lipids and enzymes and the interactions, they all can have their own literature references, so that you can actually look up specifically where that knowledge is coming from. Particularly interesting if that knowledge is not matching with your data, and the interoperability is also done, like in the other presentations, with identifiers. And the thing here is this cross references to other databases. That is the kind of thing that you need if you want to integrate data from other sources as well. And just some examples here, and we will see that a bit more, because this is actually where Wikidata comes in, which is a source for a number of these identifier mapping things. Now, we saw earlier in earlier presentations some issues with names of lipids. The same problem we have with identifiers for both metabolites and lipids and classes of things. So long chain fatty acid, for example, is not a single lipid, but actually a whole bunch of lipids. And some databases, that representation that I mentioned earlier, have multiple identifiers for biologically the same compound, like the example here, where the deprotonation actually causes a different identifier. So these are two kinds of things that you need to overcome. But using the approaches like the ontologies, as we saw a couple of examples here as well, those approaches you can use for pathway and network analysis with WikiPathways too. Now, the problem with identifiers is that we have multiple databases, and you need to connect all that together and to reflect some of the diversity in identifiers for small molecules in WikiPathways. You see an overview here. There's lipid maps down there. There's CABI, HMDB, and a good number of compound identifiers. That is nice from the perspective that it really gives a lot of flexibility of people drawing these pathways and capturing the uncertainty. So if you have done an experiment that you do not know the stereochemistry, well, that's something that you can specifically then annotate in that pathway by carefully measuring or carefully choosing your identifier. Some time ago, Denise started drawing a number of pathways in collaboration with Egon. Well, actually, I mean, he drew a number of pathways, and Denise continued that work. And we have a good number of lipid pathways already here. And I just realized this is actually a screenshot from the lipid portal from some time ago, and the EpiLipidNet logo is not there yet, but on the website it is. So you can go to lipids.wikipathways.org and actually see which pathways we have there right now. And in a couple of projects, we're going to further extend this pathway knowledge about lipids and annotating things with more accurate literature, etc. So this is one of the pathways that you can find on this portal. And you can see here the enzymatic conversions, the enzymes that are involved in that, and well, you actually. So I was lucky that I picked this one, I guess, because we see the ELO-VL enzymes here that we saw in the earlier presentation too. That box on the right side, again, that's the link out to other databases, which provide you with a lot of information. And more recently, we started adding the link to Wikidata as well. And I will show in a later slide why that is so particularly interesting. Oh, okay, actually, pretty soon next. So Wikidata is is a sister project of Wikipedia. It's a global community-driven portal of a website like Wikipedia. And like in Wikipedia, there is actually quite strong chemistry project going there. People that understand the chemistry and doing a lot of really useful things there. The basic and that is changing, but the basic nature of an instance of a lipid there is pretty much driven by the INCHI and the INCHI key. So that makes it very easy to integrate additional databases into this database, or the links out to those databases in particular. Wikidata also has a very strong link to Wikipedia itself, their sister projects, and there are side links, so you can easily actually look up a lot of information in Wikipedia about compounds as well. That needs continued curation, and that is something that that we can easily do with Wikidata, because Wikidata has a strong application programming interface, so we can query it, we can make edits all in an automated way, and this is actually something that is happening a lot. So the content there, so we find a lot of lipids in there that we will see in a moment. We find Reactome and WikiPathways is in there. There's links out to a lot of different databases, and I'll give some numbers about that in one of the later slides. And Scholia, that's what we see on the right side, is a graphical user interface that started some 2-3 years ago to visualize data in this database, but focusing on, well, as the screenshot says, it started actually out as scholarly profiles. But basically what it does is link information together. And in particular, it links researchers, research institutes, articles they write, and the topics that these articles are about, and the whole information that we know about those topics that is now available in Wikidata. So also an old screenshot, I could have picked a lipid, but the idea is exactly the same. Here we have the acetic acid with synonyms here, different languages, because like Wikipedia, it's multilingual. And if we scroll down, then we find the chemistry bits. And I already mentioned, well, this thing is very INCHI oriented for the specific compounds. We find the chemical formula there, the smiles and canonical smiles or anisomeric smiles, or often actually both. The INCHI key here. But another really important thing here in Wikidata is that every statement here can have one or more reference. So more and more people are annotating there with where information is coming from. So here, for example, for acetic acid, we find that the standard INCHI here was actually retrieved from PubChem. We have the link out to PubChem. We even have some information about the page it was retrieved from, that it was English, and that the title of that page was acetic acid, and very useful when that reference was added. So when this statement was confirmed to be accurate. The smiles itself actually links to the CDK, the Chemistry Development Kit Depict website, so you can look at the structure, which sometimes is useful to identify if stereochemistry is missing or not, something that the CDK Depict can nicely do. Not relevant to this particular molecule, but in general, I've used that a lot. There's also a lot of other information, and physicochemical properties. That is something that has had my interest a lot. And here we see the boiling point for, I assume, acetic acid as well. I'm not entirely sure. And we see actually multiple boiling points here. Besides the references that I just discussed, where the accuracy might be different, we can actually also add qualifiers to the statements in Wikidata, which is done here for the pressure at which the boiling point was measured. So turning to Scholia, that is a project that was started. It actually resulted from a discussion on Twitter. That's something that I'm really happy about. Twitter is awesome in that sense, if you follow the right people. And we started, adding a visual layer about these things. So on the previous page, we only had information about acetic acid, but no information, for example, about the articles about acetic acid and when they were written, when the information was found, and so on. And Scholia is doing exactly that. And then we get visualizations like this. So for compound classes like fatty acids, we can visualize the hierarchy. For each compound with an INCHI key, we can look up a similar INCHI key focusing on the first block of the INCHI key, and therefore giving other entries in the database with the same basic structure, but with different isotopes of different stereochemistry. Links out to other databases. We actually have a collaboration with the Chemical Abstract Service, the Common Chemistry Database, which released about a month ago 500,000 CAS registry numbers as open data to the world. And me and others, we have been collaborating with them and actually annotating CAS registry numbers in Wikidata, whether they're correct. And there are about 130,000 CAS registry numbers in Wikidata now, where we have official confirmation that that is the accurate CAS registry number. This slide is for the INCHI key lookup, and what you can see here actually is that not just the charge states differences, the bottom row here, and acetic acid, where one of the carbons is a C13 or a C14. All information that we can look up taking advantage of this. This slide shows the literature link for these things, so the physicochemical properties, the references, so we can visualize that. We also can easily make a plot of the number of publications about one of the chemicals, and via that publication you can actually look up who is studying this compound, who is studying this lipid, who might tell me how to measure this property or confirm the identity of a lipid. So this is what makes Wikidata and Scholia interesting, because it will tell you a lot more about all the other information that has been ever discovered about a particular lipid. Now, a good part of the lipid map hierarchy actually is in Wikidata, and in the bottom right, the w.wiki link, you can actually try that in your browser, and you will get this hierarchy. This is not entirely perfect, and not the full hierarchy is in there at this moment yet, but we're getting pretty close. The other thing is that we're discussing that the hierarchy actually will change in nature a bit, as something that we'll update Wikidata to reflect that. So the nice thing about that actually is that we can not just ask for the literature about a specific lipid, but we can also actually ask all articles about the subclass of lipids. And here is some breakdown of the various subclasses in the lipid maps hierarchy, and the number of specific lipids or specific compounds in Wikidata for that. There was a question and some attention to the taxonomy, in which species are lipids found. Well, there's a growing amount of literature, a growing amount of data in Wikidata actually that specifies this. This lipid has been found in this species, and in this article, this link was reported. We actually have a project ongoing. The preprint is on one of the next slides, where they look at natural products. So these are not just lipids, but a lot of other compounds as well, all getting donated into Wikidata. But there is a lot of different, a lot of collaborations with Wikidata ongoing. I mentioned Reactome, Uniprot is there as well, WikiPathways, and here, for example, we have some other things. So at this moment, there are about 18,000 lipid map identifiers in Wikidata. And again, the w.wiki/link, those are the things that run the query against Wikidata. So you can actually run that tomorrow and see if the count has changed. So that is about 18,000 out of the 23 or 45,000 identifiers that there is, because I cannot guarantee that there are not computational lipids in Wikidata. I do not know that, how I can see that, if I can see that from the identifier or not. Almost 3,500 of those have confirmed CAS registry numbers. Of that, 1,200 of those lipid map identifiers have associated mass spectra from the mass bank database. There is one lipid that has actually 85 different mass spectra associated with that lipid. Similarly, there are 488 lipids with a lipid map identifier that actually have a matching PDB crystal structure. So you can actually look at what how it behaves in a biological environment, if you like. Crystallography, crystal structures are a bit more complicated, of course. And about 4,000 different lipids, it has data to which taxon it links. And here, actually, there is one lipid that is linked to 1,800 different species. Each one of them linked to or supported by an article. This is that preprint that I just mentioned. This is a collaboration to which I contributed from my Wikidata experience. But Adriano and Maria, they did the massive amount of work behind this new database that we will see soon online. Here is a poster that says a bit more about Wikidata and Scholia, that provides some information, and in the middle there, a bit small, you can see identifiers, and you can see a whole list of databases that it links out to. And with that, I have a run through, and I don't know how I'm doing in time, but here are a couple of pointers to educational resources. So I'm not sure how much time we have before we start the Q&A, so this, for example, is an academy, and a series of small tutorials that you can walk through in a number of different a sequence of different tasks about working with Wikidata. So, for example, a walk along a pathway. I would skip the biology 101 and I just start with the green one. Or if you're more interested in actually how are these pathways created, how do we draw them, we have the blue area. And these things give a lot of information about how do we represent the biology in these pathways. At the very bottom, that's also very interesting, particularly for our future lipid pathway knowledge base effort, is actually all the curation, curation that we do on these things. So if we want to get these pathways really the way we want it, with the right kind of information and the knowledge of where does this information come from, how certain are we about that, that's what the Q&A protocols are for. In the gray at the bottom, that's a general curation protocol that we have, but we have more specific for certain for certain communities that we collaborate with. This one is a metabolomics data analysis practical. Basically, it does the same thing as the other presentations, but this shows how we do this for PathPhysio. So if you want to do this a pathway enrichment analysis, then go to this practical. It's a general metabolomics practical, but the idea is the same. You have your identifiers for your lipids, and that is what you link to your knowledge base. So it should actually be compatible with a number of the tools that we saw earlier. This is a slide that I added during the session earlier. These are two R packages that you might find interesting. On the left side, the pathway analysis, that's the R WikiPathways package. So if you're into pathway pathway analysis and network analysis with Cytoscape, then the R WikiPathways vignette or package. Actually, the package has 4 vignettes, I think. That's the package to look at. And if you are more, if you have identifier problems, then BridgeDB might actually be the R tool to look for. Except if you have these the identifiers that we saw earlier with the common names. There, BridgeDB at this moment doesn't have a solution yet. And finally, I want to mention this Scholia, tutorial, which is about Scholia and how you link articles with topics. So if you're interested in actually having your own research annotated in this database, then here you can actually. Basically, the main subject is sort of like the keywords that you provide with your article, but you have a lot more flexibility and you can link as many main subjects to your article as you like, and specific lipids, for example. The other way around, also, this is one way that allows us to give an overview of what is the literature about this specific lipid or this class of lipids. So yeah, I will share a link to the slides so you can look at them later. These are some of the work that is behind it. There's a lot of cheminformatics and Wikidata and WikiPathways work behind this. So plenty of people to acknowledge with. Oh, big thanks basically to Open Science, and this is the group where I'm working, Big Cat. So if you want to know more about Wikipathways and PathPhysio in particular, then go to our page.